All right, guys, it is 10 o'clock, so we're going to get started today. First of all, I want to thank you guys for being here. Please remember to mute your microphone and turn off your webcam. That just makes Google Meet work better. Feel free to unmute anytime you have a question. We are recording this session, and that recording will be posted to the A+ College Ready Science YouTube channel before the end of the day. We'll do this again next week for our Week 7 review, and that'll be at 10 o'clock on October the 8th. Today, we're going to just do an overall review of Unit 2, which is about cells and cell transport. Some of this content we've covered before. We'll try to cover it all again and make sure that you have a good understanding of everything you need to know for Unit 2. So let me get rolling. So first of all, we talked maybe 2 weeks ago that there's really two kinds of cells on Earth today: prokaryote or prokaryotic and eukaryotic. And scientists think that the prokaryotic cells came first, that they evolved first. These are things like bacteria and archaea. They're very small, very simple cells. They lack a membrane-bound nucleus, and they lack all the membrane-bound organelles that eukaryotic cells have. Scientists think that eukaryotic cells evolved from prokaryotic ancestors, and that one of the ways that that happened was through the idea of endosymbiosis. We call this the endosymbiotic hypothesis, that bacteria actually phagocytized or took into themselves, essentially like almost ate, smaller bacteria. So you have small prokaryotes living inside bigger ones. And like I said, probably the initial intention was to eat it, but the smaller ones lived inside the bigger one, and it turned out it was a good thing, and that both partners in that symbiotic relationship benefited. Maybe some of the first ones taken in were small aerobic bacteria that could do aerobic cell respiration. So they provided the bigger cell that they were living in ATP. Maybe some of them were photosynthetic bacteria that could do photosynthesis and provide sugar, another energy source. Maybe some were little bacteria with a ability to move, and they almost became like a flagella or a cilia for the cell and helped it to move. So the idea is that they started living together, that this was very beneficial, and after some gene swapping, or what we call gene annealing between the different symbionts, that they became so dependent on each other that they couldn't separate, and that these small little bacteria living inside the bigger one essentially became organelles. There is some evidence to sort of back up this idea, in that mitochondria and chloroplasts both have their own DNA. It's a single circular piece of DNA that looks almost identical to that in bacterial cells. They have their own ribosomes, the mitochondria and chloroplasts do, which are almost identical to those found in bacteria. They're about the same size and shape of normal bacteria. And they have a double phospholipid membrane, which indicates that maybe they had been invaginated or phagocytized and surrounded by an extra membrane early on. One other thing that mitochondria and chloroplasts can do is that they can reproduce separately from the cell that they're living in. And the way they reproduce is almost identical to prokaryotic fission, or what we call binary fission, which is the way bacteria reproduce. So again, scientists think that prokaryotic cells evolved first on Earth, and that the eukaryotic ones evolved from them, and that one of the main ways this happened is through endosymbiosis, or the endosymbiotic hypothesis. Another really important topic in this cell unit is the importance of surface area to volume ratio, which we might notate like that. The surface area, think of that like the cell membrane of a cell. The surface area's job, the cell membrane's job of a cell, is to transport things in and out of the cell. Think of the volume like the internal parts of the cell. As the cell gets bigger, the volume increases proportionally faster than does the surface area. So bigger cells have a lower, so we'll say bigger cells have a lower surface area to volume ratio. Well, what that means is it becomes harder and harder for bigger cells to transport materials. Cells need a high surface area to volume ratio. And one of the main ways to maintain that is just to stay small. And that's why almost all cells are tiny, tiny things. And even big organisms are made of just lots and lots of tiny cells. On your AP Biology formula sheet, there's a pretty comprehensive list of formulas for calculating surface areas and volumes of different shapes. I wouldn't be surprised to ask for them to ask you on the AP exam to calculate the surface area of a cell, calculate the volume of a cell, and then calculate the surface area to volume ratio of that cell. For example, let's say we were doing that for this cube right here. We're saying that cube is 3 centimeters on each side. So to calculate the surface area of a cube, you're going to use this formula. So that's 6 times side squared. So the sides are 3. So we'd say 3 times 3. Get 9. 6 times 9 to get 54. And that would be 54 centimeters squared. To get the volume, a cube is a rectangular solid. Length times width times height. So we're going to go 3 times 3 times 3. And 3 times 3 gives us 27 cubic centimeters. So our surface area to volume ratio is 54 to 27. It's fine to write it that way. Typically, though, it's reduced, almost like a fraction is reduced. And the easiest way to do that is just to divide both sides by the smaller of these two numbers. So let's divide both sides by 27. So 54 divided by 27 is 2. 27 divided by 27 is 1. So that's the reduced version of the surface area to volume ratio. I want you to note that the 2 centimeter cell has a 3-to-1 surface area to volume ratio, and the 1 centimeter cell has a 6-to-1 surface area to volume ratio. That means this small cell is much better at transporting than the 2 centimeter one, which is much better at transporting than the 3 centimeter ones. Cells stay small so that they can maintain a high surface area to volume ratio, and therefore they can transport nutrients and oxygen in and waste out. There are other ways, other adaptations to increase that surface area to volume ratio. One is to be sort of a long and skinny cell, and another is to have surface bumps like that. For example, the internal surface of your small intestine, those cells have these little projections called villi and microvilli. And those guys increase the surface area without really increasing the volume very much. And that makes those cells really good at absorbing nutrients from the internal part of the small intestine. So a high surface area to volume ratio is critical for cells, and it's critical so that they can transport things in and out of the cell. All right, so let's talk about the membrane of the cell, the plasma membrane. A lot of people just call it the cell membrane. We know that it's made mostly of a phospholipid bilayer, which we see here. These phospholipids, remember, are what we call amphipathic molecules. And that means they're both polar and nonpolar. They have a head, which we see here in red. That's polar. It's charged, and it's charged mostly because of this phosphate group. If you remember from regular chemistry, the phosphate group is PO4 minus 3. So it's a very charged ion. And then the other side is made of carbon and hydrogen tails called fatty acid tails. And this side's nonpolar. So when we put phospholipids in water, they congregate themselves into a bilayer with the nonpolar tails facing inward toward each other and away from polar water, and the polar heads pointing both out and in toward the water, because polar is attracted to polar. So polar heads attracted to polar water, nonpolar tails attracted to nonpolar tails and repelled by the water. Now, the membrane, the current model that we have of the cell membrane is known as the fluid mosaic model of the membrane. It's called fluid because the phospholipids are liquidy. Think of them almost like a slightly thicker version of like vegetable oil. The mosaic part is because there's all kinds of things that are embedded within that phospholipid bilayer. We see a lot of things in this picture that are blue. These are all proteins. So there's lots of proteins embedded within the membrane. You see this really bright yellow molecule. This is cholesterol. Cholesterol is embedded in there to help the membrane maintain its liquidity, and also to keep it from being too liquidy, to help it from being too runny, to give it a little bit of thickness. Let's talk a little bit about some of these proteins that are embedded. Lots of them are for transport. So there are some proteins that run all the way through. We might refer to those as channel proteins. And they often help a cell to transport in a very specific substance, like an ion or a large molecule, typically through the process of facilitated diffusion, which we'll talk more about here in just a minute. There are other proteins called carrier proteins. It's hard to figure out which one of these is a carrier. Let's just say that this one right here is. These molecules carry out the process of active transport. They actively, which means using energy, pump substances either in or out of the cell against the concentration gradient, from low concentration toward high concentration of that substance. There are other proteins that have sugars attached to them. These are sugars right here attached to these proteins. These often act as recognition proteins. So sometimes we might refer to these as like the name tags of the cell. They're important for identifying your cells to your own immune system so you don't attack it. These glycoproteins are also often involved in cell-to-cell communication. And we'll talk more about that in Unit 4. All right, let's move on just a bit now. So we know that eukaryotic cells have all these membrane-bound structures inside. They're surrounded by a cell membrane. Some actually have a cell wall outside that membrane for support and structure. One of the main differences, though, between a eukaryotic cell and a prokaryotic cell is that the eukaryotic cell has a membrane-enclosed nucleus. Essentially, it has DNA that's surrounded by a nucleus and separated from the rest of the cell. The liquidy part of the cell is the cytoplasm or the cytosol. And then we have numerous membrane-bound organelles. And we're going to talk more about these organelles on this next slide. So these organelles allow the cell to compartmentalize, which essentially means it divides the cell into little pieces, pieces that become specialized, take on a very specific job, become very efficient at it. Thus, that increases the overall efficiency of the whole cell. Think of compartmentalization in a cell sort of like the work that happens on an assembly line, where every person on the assembly line has a very specific job that they become good at, fast at doing. Probably nobody on that assembly line can build a whole car by themselves, but by doing each piece separately, each task separately, the process becomes very efficient and the cars are made quickly and made well. That's what compartmentalization allows to happen in a cell. It allows for specialization, division of the labor, dividing up the work, and it increases the overall efficiency of the cell. So organelles are about compartmentalization. So we're not going to talk about all the organelles and what they do today, but we are going to talk about a group of organelles that work together. Those organelles are known as the endomembrane system. So this typically starts at the rough ER. Remember, the rough ER is called rough because there are ribosomes on the surface of it. And we see the rough ER right here. The ribosomes make proteins that go down into the rough ER where they're modified a bit and eventually packaged in a vesicle, which we see here. We can call those secretory vesicles or transport vesicles. Typically, those vesicles then move to the next part of the endomembrane system, which is the Golgi apparatus. You might hear it called the Golgi body or Golgi complex. The vesicle becomes part of the Golgi apparatus. While there, the proteins are modified. They may have sugars or lipids added to them. They're also stored there for a while until they're needed. Eventually, though, the protein will work its way through the Golgi apparatus and will break off in another transport or secretory vesicle, which we see happening right here. That vesicle will then move toward the cell membrane. That vesicle is made out of phospholipids, so when it bumps into the cell membrane, they fuse. The phospholipids in that vesicle actually become part of the cell membrane and help it to grow. But the proteins or lipids that were inside that vesicle are either pushed out in a process called secretion or exocytosis, and those can then go to other cells or in your body's fluids. Or some of those proteins may actually become embedded in the cell membrane and work as carrier proteins or channel proteins or recognition proteins. So all those organisms together, rough ER, transport vesicle, Golgi apparatus, transport vesicle to cell membrane, those make up the endomembrane system. The smooth ER can also play a role here by, instead of putting proteins inside the vesicles, the smooth ER will oftentimes put different kinds of lipids inside those vesicles, transport those in a transport vesicle to the Golgi apparatus, and eventually out of the cell or into the cell membrane. So again, you see this is like an assembly line. Each part has a specific job. It's really good at it. And then it passes on the product of its work to the next part, which eventually ends, in this case, with something being secreted from the cell so it can be used by other cells nearby. All right, so we're going to, for the rest of our time today, we'll be talking about different ways cells transport things. And first, we're going to talk about what are called passive modes of transport. They're called passive because they don't require the cell to use its own energy sources like ATP. No ATP required in this case. Now, for things to move, there still has to be energy involved. For most passive modes of transport, the energy that causes the movement to happen is just heat from the environment, or body heat in this case. There's three main types of passive transport that we talk about in biology. One of those is simple diffusion, and sometimes we just call it diffusion. We know that's when things move from where there's a high concentration of that substance to where there's a low concentration of that substance. Osmosis is the diffusion of water. We're going to talk a lot more about it on the next slide. And facilitated diffusion is the movement of things like ions and large molecules that have to occur through a protein. These things typically can't go through the phospholipids. There has to be another way in. So it's diffusion with help, which is what facilitated means. And that help is typically provided by some type of protein. For example, most water gets into the cell through little channel proteins called aquaporins. So most water moves into the cell by a process called facilitated diffusion. All right, so let's talk about osmosis, because this is the one that's the most confusing for folks, really because we define it in 4 or 5 different ways. The most basic definition of osmosis is it's the diffusion of water, and it moves water from areas of high water concentration to areas of low water concentration. Oftentimes, though, when we talk about osmosis, we talk about it not so much in terms of the water, but in terms of the things that are dissolved in the water, the solutes. So when we talk about it in terms of solutes, water moves from areas of low solute concentration, which is another way of saying that the solution is mostly water, to areas of high solute concentration, which is saying to areas where the water concentration is low. Note that these two definitions say the same thing. This one just says it in terms of the solutes. This one says it completely in terms of water. Oftentimes, too, we use these two words, hypotonic and hypertonic. Hypotonic means a solution that has very few solutes, a low solute concentration, and therefore a high water concentration. And a solution that's hypertonic has a high solute concentration and therefore a low water concentration. So we can also define osmosis by saying it moves water from hypotonic areas to hypertonic areas. Osmosis is most important when there's a membrane that's impermeable to the solute that we're talking about, but permeable to water. That's when osmosis is really important. When there's a solute either outside or inside the cell that can't move through the membrane easily. So therefore, to even out concentrations, water moves either into or out of the cell to do that. Another really important topic in this unit is water potential. Water potential really measures water's ability to move. It's abbreviated by the Greek letter psi, which looks sort of like a trident, because this is water, and you guys know that Poseidon is the Greek god of water, and he carried a trident. So here's yet another way to define osmosis. Osmosis moves water from areas of high water potential to areas of low water potential. What's important about water potential is it takes into account the solutes, like all those other definitions that we just talked about. But another thing that's important for moving water is pressure. This becomes really important for cells that have walls, like plant cells, fungal cells, bacterial cells. So water potential takes into account both of those things, the solutes and the pressure. Oftentimes we'll see this psi have a T on it. That means the total water potential is equal to the pressure component of the potential plus the solute component of the potential. This formula is on your AP Biology formula sheet. So it's one that you need to know how to use and expect to see it on the AP exam. Here's another example of why this is important. Let's say we had a plant cell right here in pure distilled water. This means no solutes. Distilled water is just water. Inside that cell, there's lots of solutes, sugars, salts, other kinds of solutes. Let's assume that those solutes can't get out of this plant cell. If that's the case, if the cell membrane of that plant cell is impermeable to the solutes but permeable to the water, water starts to move in from areas of low solute to areas of higher solute inside the cell. And according to our first 3 or 4 definitions of osmosis, that would continue to happen forever. But we know that doesn't happen because as more and more water moves into this cell, because there's a wall there, more and more pressure starts to build up. So as more water goes in, the pressure inside that cell goes up and it's pushing outward. Eventually, there's so much pressure that even though the solute component of this water potential wants to keep water moving in, the pressure says, nope, I don't think so. That's all the water that can come in at this point in time. So water potential takes into account both the solutes and the pressure, which are very important for any cells that have cell walls, which remember are plant cells, fungal cells, and also most bacterial cells. So just a couple of things to remember about water potential when you're doing calculations. The water potential of pure water, the total water potential of pure water in an open container is said to be 0. And this happens because there's no solutes and because the only pressure that's affecting that water is atmospheric pressure. The atmospheric pressure is due to the air around it. In most cases in AP Biology, the pressure is 0, unless we're talking about a cell with a wall that has water moving into it, increasing that pressure. Oftentimes in AP Bio, you're going to be asked to calculate the solute potential. This formula for solute potential is also on your AP Bio formula sheet. Let's talk about this formula. So this is the solute potential. It's equal to negative I CRT. I is called the ionization constant. And it essentially looks at what happens when something dissolves. Here's what you really need to know for AP Bio about the ionization constant. If your solute is a sugar, when sugars dissolve, they don't ionize. So if your solute is a sugar, I is always equal to 1. It doesn't matter what sugar you're talking about. The other kind of solute you're going to deal with, though, in AP Bio are salts. When salts dissolve in water, they break up into their individual ions. For example, the most common salt, sodium chloride. When it dissolves, it breaks up into sodium ions and chloride ions. The important thing to know, though, is that one formula unit of NaCl, one formula of that, breaks up into two formulas of ions, two formula units of ions, or 2 moles of ions. So I for sodium chloride is 2, because one of these breaks up into 2 different ions. Now, it could get confusing, and I've never seen them do this on the AP exam, but that doesn't mean they might not. Let's say we had a solute like aluminum chloride. Well, when that ionizes, that breaks up into an aluminum ion and 3 chloride ions. So what that means is that one of these separates into 1 plus 3, 4 ions. So I for aluminum chloride would be 4. Point being, what you've got to know, for sugars, I is always 1. For salts, which remember are made of a metal and a non-metal connected together, I is equal to the number of ions that ion dissolves in, or ionizes into when it dissolves in a solution. In almost all cases, in all cases I've ever seen them put on the AP exam, for salts, the salts they've used are something simple like NaCl, which typically is going to break into two ions. In this case, Na plus and Cl minus. So for most salts that you're going to see in AP Bio, I is going to be 2. For all sugars, I is going to be 1. The C in this formula is the concentration. So this is the concentration of solutes that you're given in the problem. And that C is going to be in terms of molarity. R is called the pressure constant, and it is always this value right here. And that's going to be given to you on your AP Bio formula sheet, which you can use throughout the exam. So you don't need to memorize R, it's given to you. Notice it has some strange units on it, liter bars over moles times K, but you've got that, it's given to you. The temperature is T. That temperature has to be in Kelvin. And they almost always are going to give you the temperature in Celsius. So you're going to need to be able to convert Celsius temperatures to Kelvin. And all you need to do to make that conversion is add 273. So if you had a Celsius temperature of 20 degrees C, you add 273 to it, and now you've got 293 degrees Kelvin. For this equation to work, the temperature has to be in Kelvin. Make sure you know that. This whole section is straight off of your formula sheet, so it tells you on the formula sheet essentially that you need to do that. Let's look at just a couple of quick problems. So if you were here last week, we worked similar problems, but these are slightly different than the ones we worked last week. So here's the molar, we've got the molar concentration of this, in this case, KCl. You first have to realize, hey, this is a salt because it's made of a metal, potassium, bonded to a non-metal, chlorine. It's in a container. The concentration of the solution is 0.6 molar. The pressure of the solution is 0. What is the total water potential at a temperature of 20 degrees Celsius? This is our equation for total water potential. We've been told that this part of the equation, the pressure, is 0. So to get the total water potential, we've got to calculate this part, the solute potential. The solute potential is equal to negative I CRT. Let's first figure out the ionization constant, I. So let's take KCl, a salt. When it ionizes, it breaks up into its ions, which are those 2. So one formula unit of this breaks up into 2 units of ions, which means I equals 2. So negative 2 times the concentration, which is 0.6. That's the molarity. I like to write that big M, molarity, as what it stands for, which is moles per liter. And I'm going to do that so I can show you what happens with the units on this problem. So now we need to multiply that by the R, the pressure constant, which is right here, 0.0831 liter bars over moles times K. And then we need the temperature. So our temperature is 20 degrees Celsius. Remember, we need to add 273 to that. to give us 293 degrees K. Let's cancel out units. So K's cancel, moles cancel, liters cancel. We're left with bars. Let's multiply. So let's go 293 times 0.0831 times 0.6 times 2. And let's don't forget this negative sign out in front. It's really important. So we end up with a water potential of negative 29.2 bars. That's the potential inside the cell. No, I'm sorry. That's the potential inside the solution. So that's out here in the beaker. The next part of this question says, if a plant cell with a total water potential of -4 is placed into a beaker of the solution described above, will water move into or out of the cell? Assume the cell's membrane is impermeable to the solute. Justify your answer. So inside the cell, the water potential is -4. Remember that water always moves from areas of higher water potential to areas of lower water potential. Some of you are going to look at this and say, well, 29.2 is bigger than -4, so water's going to move in. We're dealing, though, with negative numbers. Remember that -4 is closer to positive than -29 is, which means -4 is the bigger number. -4 is greater than -29.2. Therefore, in this case, water's going to move from higher water potential, -4, in the cell, to lower water potential, -29.2, outside the cell. So water's going to move out of the cell, and it's going to do that because water always moves from areas of higher to lower water potential. And that's really what you need to say to justify your answer. So let's look at one other problem here. So this one, in this case, looks very similar problem. And this time, though, the solute is glucose. Well, the name ends in OSE. Hopefully that tells you it's a sugar. And remember, when we're dealing with sugars, I equals 1 for all sugars. So let's calculate, first of all, the total water potential. Here's our formula. They're telling us again that the pressure here is 0. So to find the total water potential, we've got to find the solute potential. So let's go back to -I CRT. We just figured out that I for this problem is 1. The concentration is the molarity, 0.2. Remember, that's moles per liter. That's what big M means. The R is the pressure constant, 0.0831. And again, the units are liter bars over moles times Kelvin. And then the temperature is 273 plus 30. So we got to add 273 to 30 to get 303 Kelvin. Cancel our units. Everything cancels out except bars. And then let's do the math. So let's go 303 times 0.0831 times 0.2 times 1. And don't forget that we got that negative out front. So our water potential comes up to be -5.04 bars. That's the water potential out here in the beaker. In this case, the water potential inside the cell is -12. And we want to know what will happen to the cells in total internal water potential as time passes. Well, before we answer that, we've got to figure out what happens with water. Does water move in or out? And remember that water always moves from higher water potential areas to lower water potential areas. So water's going to move in. So this cell is going to gain more and more water. So let's think about the water potential inside the cell for a minute. And we've got to think about our two formulas as we do that. As more water goes into that cell, the pressure in the cell goes up. So this component goes up. As more water goes in, this concentration gets higher. The concentration gets lower. And because this is a negative number, that causes the solute potential also to increase, because a smaller negative number is actually a smaller magnitude negative number is actually a bigger number, right? So because the concentration of the solutes decreases, the solute potential also increases. So if both parts of the total water potential equation increase, that means that the total water potential will increase. So our answer to this question, water potential will increase, and our justification is going to be because the pressure inside the cell is going to increase, and because the concentration of the solute is going to decrease, because that solute's getting diluted, which causes the solute potential to go up as well. All right, I've got one more sample that I want us to quickly look at. So here we have a cell membrane separating two sides of the cell, inside and outside, side A and B. And first of all, the triangles represent solutes. And we're told that on side A, we're starting with a solute potential of negative 1.7, pressure of 0. On side B, solute potential of negative 0.6, pressure potential of positive 1.6. And we want to know if the volumes of the two sides are initially equal. So let's say this is where the water starts out. In which direction, first of all, will water flow? Remember that water flows from areas of higher water potential to areas of lower water potential. Over here, the total water potential is negative 1.7 plus 0. So that's the total water potential on this side. Over on side B, the total water potential is negative 0.6 plus positive 1.5. That gives us a water potential on this side of 0.9, positive 0.9. So the water potential on side B is higher than the water potential on side A. So the water is going to move from B to A. That's our answer to number 1. We've already calculated the total water potential on each side. Side B, positive 0.9. Side A, negative 1.7. Which side is hypertonic? Remember that hypertonic means more solutes, higher solute concentration. Well, that's side A, more solutes. Side B is hypotonic. It has less solutes, a lower solute concentration than what we're comparing it to, which is side A. What will happen to the volume of side A? Well, in number 1, we determined that water is going to move from B toward A. So as that happens, the volume on side B is going to go down, and the volume on side A is going to go up. So side A volume will increase, side B volume will decrease, and the justification is that water is moving from higher solute concentration, or higher water potential rather, on side B to lower water potential on side A. And number 6, why is the initial solute potential on side A lower than the initial solute potential on side B? Well, we got to go back to our formula for solute potential. I is the same on both sides. R is the same on both sides. The temperature, we're assuming, is the same on both sides. So what's different is the concentration. The concentration on side A is higher than it is on side B. And because this is a negative number, that negative is out front, that actually makes the side A calculation for this be lower than on side B. That negative number makes the higher solute concentration turn into a lower number on side A than it is on side B. All right, so let's end up today by talking about, first of all, water potential and how it relates to transpiration. We talked about this in our first discussion of water potential last week. In the soil, the water potential is somewhere around negative 0.3. It's a little lower in the xylem of the roots, the water-carrying tissues in the roots. So water moves from higher to lower water potential. Water goes into the root xylem. Well, up in the trunk, the water tubes, the xylem, have even a lower water potential. So water moves from the root xylem into the trunk xylem. Up in the leaves, the water potential is even lower. So water moves from the trunk xylem into the leaves. In the air spaces of the leaves, it's even lower. So water moves from the cell walls of the leaves into the air spaces, and those eventually lead to the stomates, the tiny pores on the bottoms of the leaves, which connect the leaf to the outside air, where the water potential is even lower. So water moves out of the leaf, out into the air, and this process moves water from areas of higher water potential down here in the soil, and I'm going to abbreviate that WP, to areas of lower water potential, which end up being outside air. This process moves water from the ground up through a plant, even a tall tree, and ultimately back out into the air. That's transpiration. All right, the last thing we'll mention today is active transport. So active transport are types of movement, cell transport mechanisms in plants that require the cell to use their own energy. Typically, they're using ATP. If we're moving small things, we oftentimes just call that type of cell transport active transport. This uses carrier proteins, and it typically pumps substances, usually ions or things like that, from low concentration to areas of higher concentration, which is not the way nature wants to do things typically. Nature is usually moving things in a way that balances out concentrations. In this case, if you're going to move things against concentration, that requires energy, and that energy is provided by ATP. Famous examples of active transport would be the hydrogen ion pumps that are involved in cell respiration and photosynthesis. There's also a pump called the sodium potassium pump that's really important to help your nerves and your brain work. Bulk transport is another type of active transport which requires the movement of large substances or large amounts of a substance. And there's really two types of bulk transport that we talk about in bio: endocytosis and exocytosis. Endocytosis is typically the movement into a cell of large materials. We divide endocytosis into two types. Phagocytosis is taking in large solid substances. This could be like a white blood cell engulfing and taking in a bacteria. Pinocytosis, on the other hand, is taking in large amounts of dissolved things. This might be a cell taking in a hormone or a cell taking in some type of solutes in a large amounts. On the other hand, exocytosis is also sometimes called secretion. So this is when a substance exports something out of the cell into the fluids that surround it, so that those substances might be used for by other cells nearby. This might be how a cell exports a hormone or exports a cell signal. We also want to talk briefly before we do. This is showing you phagocytosis. So here's a cell that's surrounding this large particle with its cell membrane. It eventually closes off these two sides of the cell membrane together. They fuse because they're both made out of phospholipids. That encloses that solid substance in a vesicle, sometimes called a phagosome. Typically, the lysosome will then bump up near this and dump into this phagosome enzymes and acids, which will digest that material inside the cell. And we call that, it's a type of internal digestion. Pinocytosis, we're taking dissolved solutes, in this case, these little blue square-like substances, surrounding them in a vesicle, pulling them into the cell. In some cases, pinocytosis is stimulated by receptors. So first, the solute has to bind to a receptor on the cell membrane, which then triggers the cell membrane to fold inward, forming a vesicle, pulling that substance into the cell. So this is really a type of pinocytosis. It's just a type that's stimulated by receptors binding to solutes. Remember that endocytosis is an active process. It requires the cell to use its own ATP. Here we see exocytosis. So this is the endomembrane system that we saw earlier. And we see that proteins are made by the ribosomes on the rough ER. They're packaged in the endoplasmic reticulum into a transport vesicle, which moves to the Golgi apparatus where those proteins are modified, eventually breaking off in another vesicle, bonding with the cell membrane, and then secreting out the proteins inside, or lipids, could be lipids. That process is exocytosis. Again, an active process. All that movement requires ATP to be used by the cell. The last thing I want to mention is co-transport. So again, this requires energy, but it doesn't necessarily require ATP to happen. This often happens when a substance like sodium, notice there's a lot of sodium outside the cell, not very much inside. So the sodium moves in through a protein, which is this yellow thing here. It moves in from high to low. It's moving with the concentration gradient. Well, that's almost like an energy source. And this protein can use the energy of those moving sodiums to transport something else against its concentration gradient. So in this case, there's not a lot of glucose outside the cell, but there is a higher concentration inside. This protein is going to use the energy from those moving sodiums, the kinetic energy of those moving sodiums, to power the pumping, essentially, of glucose from lower concentration outside the cell into the cell. That requires energy, and that energy is supplied by the kinetic energy of sodium ions moving with the concentration gradient. So the glucose can't be transported by this protein unless at the same time that protein is transporting sodiums with the concentration gradient. So that's called co-transport. A couple of years ago, there was a pretty big question on the AP exam about co-transport. So you should be familiar with it. It's when two substances are transported by the same protein, one with the concentration gradient, one against. And the energy to move the one against the concentration gradient is provided by the kinetic energy of the substances moving with the gradient. All right, that's it for our week 6 content review. Again, we'll do week 7 next week. Week 7, we'll be moving into a new unit. That unit is cell energetics. Week 7 is going to look at things like enzymes. It's also going to look at cell respiration, which is how our cells make ATP. And it's also going to include a look at photosynthesis, which is how plants and some bacteria and algae use energy from the sun to create food molecules, essentially sugar. All right, I hope to see you guys again next week. Thanks for being here. I hope it helped.